Hello, my name is Lucia Sivilotti. I work at University College London. I have spent the best part of my career working with single channels. The purpose of my talk today is to introduce you to the technique and to show you the range of scientific questions that you can address with it. From the very simple and relatively easy to do to the most difficult, as an introduction for today's talk by David Colquhoun. So, when can you get a single channel recording? In order to get a single channel recording, you have some conditions that have to apply. You have to have a high patch resistance. So that means having a small patch of membrane and to have a high seal resistance. So you have that the pipette has to be well sealed to the membrane. You can see here you have 150 megohms. This is Hamill, Martinaire, Sackman, and Sigworth. But when the suction is applied and the seal improved to 6 gigaohms, you can see how the record changes. And you can see this better at the bigger display here. And you can see how much cleaner, less noise you have here after you have established the giga seal. So you can look in Adley and Stanfield textbook to see what conditions you need to have. Single channels are typically relatively small signals, a few picoamps. The conductances, which are measured in Siemens, a Siemens is very simply the reciprocal of an ohm. The conductance of a single channel can be up to 250 picosiemens. The theoretical limit is supposed to be about almost 400 picosiemens, and you can look it up in Hille. There's a range of systems that you can use single channel techniques on, and those vary with all the configurations of the patch clamp technique. Of course, those will mostly be either isolated patches, either cell attached or excised. If you have a very small cell, such as a cerebellar granule cell, you can sometimes see single channels even in whole cell recording, but it's very special conditions. Then you can apply agonist or voltage in all sorts of conditions. They can be either steady state or concentration of voltage jumps. You can use native channels. You can compare native channels to those that you obtain in recombinant expression, or you can reconstitute channel proteins into artificial membranes. There's a range of things that you can do, but those are all based on two general measurements. One is amplitude measurements, and the other one is time measurements. The time measurements can be open probability or open and shut times. All of these measurements can be relatively simple, but you must understand how the signal is distorted by your apparatus and by the filtering that you have. In this slide, I have listed a range of questions that you can address with single channel recording. Now, these go from the easy and quick questions that you can address with pretty standard software, and those are generally based on the amplitude and the general look of the single channel openings. I have highlighted here the examples that I will be stopping on and expanding on later on. So we go into something a little bit more difficult, measuring directly the maximum open probability, for instance, of a ligand-gated ion channel, which is quite useful to pharmacologists to estimate efficacy. Then we go towards the deep end of the spectrum, towards kinetics. And here, the most difficult of these questions is establishing in detail the channel activation mechanism, broken down in the steps that make it up, and classifying them as, I don't know, concentration dependent or voltage dependent, measuring the rate constants of their transitions. And ultimately, what we're aiming to do is to establish the energy landscape of the protein. Now, this is difficult and slow, And it's difficult in the way, I suppose, I used to think crystallography is difficult. So you can spend a lot of time obtaining very high quality recordings and then not be able to fit them and get to a total success in obtaining the channel activation mechanism. So success is not guaranteed. As I promised, we start at the shallow end, amplitudes. So the first thing is, how big is the current through this protein? If the current is big, it's more likely to be a channel than it is to be a transporter. And that is an example here. This is taken from the work of Randy Stockbridge and Chris Miller, who cloned a bacterial protein, and they didn't know whether it was a channel or a transporter. The sequences resemble transporters, but they have a conductance of 7 picosiemens. So it's so high that it must be a channel. And why is a channel so much more, has a channel so much more conductance than a transporter? Well, a channel is a very special hole in the memory. And it's diffusive, you open the gate, and it's diffusive and it's fast. Now, this is a little calculation that you should try for yourself. 1 picoamp of current is 6,000 ions per millisecond. So that is a huge number of ions going through a channel. And you can see here we have, for instance, 2 picoamp currents. A transporter, on the other hand, is relatively slow. So we must close the gate at one end and open the gate at the other end. And so it's relying on a conformational flipping and flopping of the gates to let the ion, or rather the cow in this case, through. So the maximum that we can achieve with that is probably 100 ions per millisecond, which we are in the region of 0.3 over picosiemens. So that is a relatively simple use of single channel recording. Other questions that we can ask with single channels exploit another aspect of single channel recording. For instance, we can isolate the protein in the membrane patch and have relatively controlled conditions, much more better controlled conditions for the activation of the protein. One example is here shown. It's a BK channel. So how is the BK channel, a potassium channel, activated? You can see here we have the openings here are upwards, and you can see them here. This is minus 10 millivolts. As we depolarize, you can see that not only the driving force increases, and because of that, the current increases, bigger unitary currents here, but you can see also that the open probability of this patch has increased. This is a measurement that we call NP open, because we don't know how many channels are in the patch. Here there are at least 2, as you can see. But you can see very clearly that the open probability has increased as we depolarize the patch. Now, the question here that was addressed, this is a 1981 paper, pretty important paper, was whether the BK channel was activated by depolarization or was activated primarily by changes in internal calcium. And in these conditions where we have a patch that we depolarize, it's very unlikely that the internal calcium is affected by the depolarization that we have in such a small patch. So this establishes that the open probability of the BK channel increases with the depolarization of the patch. Now I'm going to show you a few examples on how we can identify channels depending on their activation or on how they look when we get single channel recording from them. These are calcium channels, voltage activated calcium channels, and you can see here this is a cell attached patch that is either depolarized from a relatively depolarized membrane potential to plus 10 millivolts, or it's depolarized from a fairly hyperpolarized potential to minus 20 millivolts. So you can see that the type of channels that we see in the two different conditions are quite different. You have either big, slowish channels, and these are the L-type channels, or you can have small, short opening, faster channels, and these open more quickly perhaps when there is the voltage jump here. If we produce an ensemble of these recordings here, we just sum them all together, you can see a slowish current, and we can see a current that has got a different kinetics for the T-type channels. So you can clarify what different voltage commands have activated. And you can see here that it's obvious that these channels are different. These are not fantastic recordings, but in terms of the noise, but you know, it makes the point quite clearly. This is another example of the pretty obvious, clear-cut result. And this is the subunit composition of the nicotinic acetylcholine receptor. Now, in fetal muscle, we find channels, here the openings are downwards, that are smaller and tend to have longer open times than in the adult. If we express in xenopus oocytes these four subunits, alpha, beta, gamma, delta, we get channels that look exactly like the fetal ones, but if we replace the gamma with the epsilon subunit, we get channels that look very much like the adult channels. So this establishes pretty well the composition of the native nicotinic acetylcholine receptor in fetal or adult tissue. Now, just in case you have this idea that success is inevitable with this sort of techniques, I'm going to show you one of my papers in which I was trying to do what Mishina and Sackman did for the native muscle nicotinic receptor. So I was taking recordings from a native autonomic ganglion. This is a neuronal nicotinic acetylcholine receptor. And I was comparing with the recordings, the single channel openings that you get if you inject xenopus oocytes with alpha 3 beta 4 subunits, or rather DNA for these subunits, which are supposed to be the major constituents of native autonomic neuronal nicotinics. And you don't need to do very much, very difficult measurements to see that these two channels are completely different. The channels in xenopus oocytes have got smaller openings that last a very long time. They run down very, very, very quickly. This is the only group of openings that we got in the outside-out patch. They're much smaller, I was saying, than the conductance of the superior cervical ganglion nicotinic, and the time of opening is completely different. You wouldn't even need to measure these. But I'm going to spend now a couple of slides telling you about measurements and telling you why we must be careful even with amplitude measurements. Now, it's pretty clear that here you need what? You need the electronic equivalent of a double ruler to measure this conductance, or rather how many picoamps there are, and then go to the conductance. But here, in these briefer channel openings, take a look. The openings don't look to be, well, there are some that are clearly a different conductance, this and that, but the openings look kind of different in amplitude. Look at the brief openings here. This seems big, this seems smaller, this seems smaller. The shorter openings look like they may be smaller than the bigger opening. Why is that? Now we must look, and we will in the next slide, look at what filtering does to the amplitude of short openings. Single channel recordings have to be filtered to be made interpretable and not look like a mass of noise, but filtering comes at a cost. And this is shown here in the top panel. Now, the opening of an ion channel is as instantaneous as may be. We don't know how long it takes for the ion channel actually to open, so what this rise time really is. But if we filter these pulses, the effect of a low-pass filter depends on how long the pulses are. So you can see that with a long pulse, we have the equivalent of that oocyte neuronal nicotinic, and we can measure, we have a detectable top here that we can measure with a double ruler. But shorter pulses that are, say, 1 time the time rise of the filter, and you will find a table below here that tells you what that is, they become smaller and smaller. They spread out, so the area is not changed, but the amplitude is reduced. So you can no longer measure them very simply, and you start having trouble measuring them also with threshold crossing methods. Now, we have a table here that shows you the numbers for commonly used filtering. I mean, for channels that are as big as the one that I've been working with, we usually work with 3 or 5 kilohertz, 5 if you have a very good recording. But you can see that with 3 kilohertz, in order to get to 90% of the amplitude of the opening, you have to have 144 microseconds of opening. And this is it's a fairly long duration of an opening as ligand-gated channels go. So we are very much in the area of measurements that are important to us. Here we're making the case for openings, but of course the same thing applies for the opposite. So for short shuttings. So the short shuttings will look less and less shut as they become shorter. And then at one end here, they'll end up mixing up and being difficult to tell from the baseline noise. So bearing in mind the caution about the filtering, how do we measure amplitudes? What sort of displays do we produce for amplitudes? We can just take all the points that are in a recording and just pop them in a program and just get a histogram. So each point here will be a point in this histogram here. The problem here is that you will get the points for the closed state. Here we have at least 2 channels opening. So these will be the points here with their noise. Then we will get some points up here for the opening of 1 channel, and some points up there for the opening of 2 channels. But you will also get some points, although not very many, when the channel is actually opening. And because it's filtered, it's not an instantaneous transition, but we will get a smear here. If you want to have a prettier picture, and it depends on how precise what is the purpose of your measurement, you can actually go one further. You can actually fit the opening and the closing of the channel with the time course that you would expect given the particular filtering that you're using. This is a time course fitting that is done by programs like SCAN, a specialist program. And then those will allow you to classify the points as shut. Then you have the transitional points that we discard, and then we have the points that are clearly open. So we can now put the shut and the open amplitudes in 2 different and see what bins we have. One final way would be simply to do scan, and that is to do time course fitting, and then that will fit not only the time course, but will give you a best fit of the amplitude of the opening of the channel. Then for each opening, you will have 1 value. And this is shown in the next slide. And here it is. So here we have the measured amplitudes, and you see the number of points is very much smaller because for each point in this histogram, which is approximates a Gaussian, it's not quite a Gaussian, we have one opening for each of the points in the bars. In the last few slides of my talk, I want to give you a first view of topics in single-channel kinetics. So let's have a look at synaptic channels and channelopathies. Channelopathies are rare conditions that are inherited, and they can be due to mutations in the receptor, in the ligand-gated ion channel at the synapse. In this case here we have muscle nicotinic receptors, and you can get congenital myasthenias. This is not to be confused with myasthenia gravis, which is an autoimmune disease. In congenital myasthenia, you have mutations in the channel. Here we see the miniature currents that are recorded from human muscle biopsy. These are patients that have these congenital myasthenia. You can get two sorts of myasthenia. These are data from Sine and Engel, who are specialists on this. And you can get, you see here, this is a control current. This is a time constant of, this arrow here is a time constant of the decay of the current. You can get a slow channel congenital myasthenia. You can see how it's got an additional time constant, very, very slow. This leads to excitotoxicity because of calcium entry. Or you can get a much faster channel decay of the activation of the channel current, and this is not enough signal to reach the threshold for an action potential. Now, this is a difficult experiment, as you can imagine, human muscle biopsy, but what you can do is sequence the receptor in the patients and then express the receptor, obtaining recombinant channels with the appropriate mutation. Now, what you can see here is interesting. These are applications, steady state at equilibrium of low concentration of acetylcholine. And you can see that in the slow channel mutant, you have longer openings. And in the fast channel mutant, you have faster, shorter openings than in the wild type. Now, this is kind of interesting because the situation here, this is an equilibrium recording. This here, the synaptic current, is a non-equilibrium event. It's a situation in which at the synapse you go from zero transmitter to millimolar concentration in microseconds, and then this concentration lasts maybe half a millisecond for acetylcholine. But you can see that some characteristics of the single channel equilibrium recording resemble those of the non-equilibrium situation. This will be expanded on when we look at dwell times and their measurements in single channels. Now, I want to finish with a look at a topic that is important for pharmacologists. And first of all, I have an admission to make. When we talk about single channel recording, we don't know that we have one single ion channel in the patch. We almost never do, for my sort of channels at least. You would need to do an experiment to count the channels in the patch, and then you would have run out of time to do the real experiment that you wanted to do. But there are circumstances in which we know that a single channel recording is from one molecule. This is a well-behaved ion channel. This is the alpha-1 glycine receptor, and this is being exposed to a high concentration of the neurotransmitter glycine. Most of the time, the channel is closed. All the channels in the patch are closed. They're desensitized, refractory. Every now and again, one channel comes out of desensitization, opens, closes, open, closes. The shuttings are very brief, very high probability of opening here, and then it shuts again and goes desensitized. and then there is another cluster of openings here. We don't know that it is the same channel as we had here, but it is a single channel. Why is it a single channel? Because we have looked, and if there were two channels working here, we would certainly see a double. Now, this is very useful because we can measure here the P open, the open probability that is elicited by a saturating concentration of glycine, a concentration of glycine that fully occupies the channel. And you can see that this is very, very high. The channel is either open or desensitized. So this tells us as pharmacologists that glycine is a very efficacious, is a full agonist on the glycine receptor. This is a pretty unique example in receptor pharmacology, that of ion channels, because this is a very difficult measurement to make. This slide, also from my work, shows you what happens when you increase the concentration of the agonist. Here, glycine, it's a heteromeric ion channel to glycine, also a well-behaved channel. At low concentrations, what we have is we have brief openings, maybe in groups of, maybe alone, or maybe in groups of 2 or 3. And these are separated by long shut times. These shut times are, I can tell you now, they are the expression of agonist affinity. What do I mean by that? The channel doesn't open if glycine is not bound. It's got a very good signal-to-noise ratio for detection of the agonist concentration. So it only opens when the agonist is bound. If the agonist unbinds, the channel shuts, and it doesn't reopen until another molecule of agonist has bound. So the length of this is the time that it takes for another agonist to bind. So it should be no surprise to you that if I increase the concentration of glycine, and this is shown here, these openings and groups of openings become closer to one another. And this is also becoming visible here. Another thing, remember desensitization? We can begin to see that we have long shut times that are desensitized, and then we have shut times between the groups of openings here that are called bursts. These shorter shut times are concentration dependent. If I increase even further the concentration, we end up in the situation that we had before, where we have desensitized shut times, and then we have very, very short shut times in there, in the clusters, and those very brief shut times and that very high open probability is an expression of agonist efficacy. This is my last data slide, and it shows you the dose-response curve that we obtain from a single ion channel molecule. With glycine, this is heteromeric alpha-1 beta channel. It's a very well-behaved channel, and you can see the open probability, and this is an absolute measurement that goes up and reaches almost 100%, as we've seen before. Now, this is the case for glycine, which is a full agonist, but with another agonist, the partial agonist taurine, no matter how high I go in concentration, this is 100 millimolar, the channel is saturated, but it cannot be kept open all the time as glycine does. Taurine is not as efficacious as glycine. This is quite an unusual sort of dose-response curves. It looks very much like the dose-response curves that you have, if you are pharmacologists, you may have obtained them, say, in a bit of isolated gut. But it is not the same thing, because here we have an absolute measurement of the open probability and of the maximum. In dose-response curves, say, in a guinea pig ileum, the maximum is hard to measure accurately, and what we measure is not a good relation. It cannot be easily correlated with the opening of channels, because it depends on how many channels you have in that particular tissue, and it may vary from one tissue to the next. And it depends on the application method for the agonist. So if you apply the agonist very slowly, you get a lot of desensitization occurring at the time that you are applying the agonist, so you get a different maximum. But this is quite a different measurement. It's an absolute measurement, and a measurement that is quite reliable and can be interpreted in terms of mechanism. The figure on my last slide is courtesy of Andrew Plested, and these are data from 2016, and it shows the number of papers published depending on how big the conductance of the channel is and how slow the channel is. So the easy channels are the ones that are big and or slow. The small channels and the fast channels are much more difficult. Thank you.